Thank you, ISMB, for allowing me to present this work to the greater community. My name is Trung. I'm a computer science PhD candidate at Duke University, advised by Alex Hardermink. And today I'm kind of excited to tell you about this transformer model that we use to kind of understand the relationship between the chromatin and gene transcription. So to start, we can kind of think of the chromatin as a combinatorial complex arrangement of proteins, histones that make up a nucleosome, polymerases, and other bound DNA binding factors that bind and unbind and move up and down the DNA and exist as this combinatory complex state that any type of DNA-dependent function will require to regulate. And for this talk, I'll focus particularly on gene transcription. So for a complex topic like the chromatin, it's easier to simplify things into a couple of simpler states. So in the first case, we have a gene, HSP26, that is not being actively transcribed. So the chromatin will look like a series of nucleosomes that are statically positioned in its promoter and its gene body. Alternatively, when HSP26 is being actively transcribed, its promoter will be occupied by transcriptional machinery, transcription factors, and will recruit polymerase, which will move nucleosomes up and down and potentially evict them from the gene body as polymerase actively transcribes HSP26. And in our lab, my colleague kind of described this yesterday, Kevin Mo-Young. We use MNase-seq, which is a factor-agnostic chromatin accessibility assay. And how MNase works is uses micrococcal nuclease, which is an endoexonuclease that cuts open DNA that is not bound and digests up to those bound factors. And what you get is this depiction of a dot plot, which represents the fragment length and the genomic position of the bound factors. So here we see a transcription factor flanked by a series of nucleosomes, and that you can kind of see that as these clusters of larger fragments and a little cluster of a smaller fragment that represents the transcription factor. So returning to our earlier example, we can see that the lowly transcribed HSP26 can be represented in this MNase-seq image as just a series of those nucleosome clusters. And alternatively, we'll see when HSP26 is actively transcribed, in its promoter, we'll see an enrichment of the smaller factors, possibly the transcriptional machinery in the promoter, and a disarray of the nucleosomes in the gene body as Pol2 actively transcribes HSP26. And we can kind of also zoom out a bit to see the fidelity of this assay. So here's the time course of HSP26. In the center is the chromatin context around the transcription start site for HSP26. And then you can also see downstream a gene that isn't actively transcribed remaining fixed throughout the time course. So this kind of gives us an idea that the biology of HSP26 is being accurately represented in this experiment. So now that we have these chromatin images, we can kind of think of them as two-dimensional representations of the chromatin state, and we can decide if there's a way to connect those chromatin state to gene expression. So the question becomes, can we use these images of the chromatin state to predict gene expression? And more importantly, if we can do that, is there a way that we can draw inferences from that model to determine what parts of the chromatin state are most salient in this prediction? To do that, we use the concept of the vision transformer. So a transformer will take a one-dimensional vector and kind of decide what pieces of what elements in the input vector will have co-relevance in deciding on the prediction of the output. And in our case, we have a two-dimensional input image. So we'll take the vision aspect of this vision transformer, which basically will segment the input images into patches and then linearize them that can be fed into the traditional transformer deep learning model. And then these transformer encoders also have this attention mechanism scheme that we can also use to roll up the attentions and link them back to the original patch that was helped inform the prediction. So put more simply, we take our chromatin input, patch and linearize it, feed them into the transformer encoders, and then feed it into a a smaller neural network, the prediction head, which will predict our gene transcript level. And once we have that, we can roll up the attention weights from the transformer encoder. So given a input chromatin image, we can take the attentions that were turned on and roll them back up and try to determine what parts of the input chromatin were salient in the prediction for the transcript level. So because we're partitioning up the input chromatin, we can kind of be thoughtful about how we decide how to patch our input. So in MNAs, we'll typically take the fragment length distribution to determine if there's a consistency across samples. So we will want this fragment length distribution on the left to be consistent. But we can also use it to decide how we segment our patches. So in this case, we'll just have small, intermediate, and nucleosome size fragments. And we can also do the same thing on the genomic position. So we want to make sure that we patch things so that we can determine the precise positioning of the nucleosomes and transcription factors, but we're also going to be limited by the computational and memory efficiency of the resolution of these patches. So the data set we'll be using is these cadmium perturbation assay. So basically, we'll take yeast, sample its chromatin and transcriptome using MNA-seq and RNA-seq, introduce this heavy metal cadmium, and then take 5 more time points following that perturbation. And what this does is that the cadmium will induce a stress response, which activates many genes, activates and represses a handful of genes, but the chromatin also will have associated changes. And we will take the first 5 time points as our training set, and we'll test against our remaining time point 120. So there's different ways that we can slice and dice the training and testing scheme, but this is what we settled on. And so with that, we can predict time point 120 with a pretty high level correlation. So half of the variance can be explained with just the chromatin alone. But more importantly, we want to know that this model is fit correctly, and then that we can examine the attentions and try to understand why it's predicted as well as it is. So in one example, we have RPS7A, which is a ribosomal subunit protein. It is repressed as translational activities are repressed under this stress condition, as it's not as important as dealing with the cadmium. And in the third row on the right panel, we see that these are the attentions that the model decides are important for the prediction. So in this case, it sees that the +2 nucleosome is the most salient feature in this prediction. And to a lesser extent, we'll see the +1 nucleosome is upstream of the transcription start site, and there are 2 important bound factors in the promoter region. And in contrast, we can take another gene. MET16 is upregulated as the sulfur pathway is required for glutathione, which is a chelation pathway for cadmium to remove it from the cell. And that we see the most important feature is in yellow, this intermediate or small fragment enrichment. And the +1 nucleosome is downstream of the transcription start site. So this attention weight idea, we can analyze genome-wide. So if I take all of the genes for time point 120 and cluster them into these 16 clusters, we can kind of determine if there are commonalities in the patterns that the attentions are binding. We can also look at the transcription levels at each of these clusters. So in these plots on the left, we have the entire time course is plotted. So the genes interquartile and min-maximum values are plotted as each of these dots for each of the time points. But only the last time point 120, is associated with these attention weights. And I'm also coloring the time course by the most highly activated most highly activated gene in the cluster. So the bottom right cluster has the most highly activated genes. And we see that there's some important features that we can contrast between the most lowly transcribed and highly transcribed genes. And also I can composite the input MNA-seq images for each of the genes in the cluster so that we can kind of get a better idea of what the attention is looking at. So looking at the lowly and highly transcribed clusters. We can see that in the case of lowly transcribed clusters in genes, the +1 and -1 nucleosomes are most informative in the prediction, while highly transcribed genes, the promoter has a very specific position in the promoter that has the small fragments that are enriched. And we can also look in the four most transcribed genes in gene clusters. We can see that this is a common pattern. The small factors in the promoter are very predictive of these gene clusters. But also, there's an interesting idea where these four clusters have some different modalities in the gene transcription. So the first two clusters, the nucleosomes look much more well positioned by, as you can see in the top right plots. And then in the second row, cluster 14, the +1 nucleosome is potentially a salient feature in this cluster. But then also in cluster 15 and 16, the nucleosomes are much more variable, but the small fragments in those clusters, they're a little bit further upstream than the previous two clusters. So potentially these positions in the promoter might be important. There might be some biological mechanisms that are driving these positions in the promoter. So it would be important to kind of think about how we further move along this analysis. So potentially these positions in the promoter will have certain sequence or motif patterns that we can further analyze, and potentially if there's nucleosome positions that are important, we can try to link them to different histone marks, or maybe the genes will have certain types of genomic features, maybe adjacent, divergent, or tandem genes upstream, or maybe at nearby origins. So we can kind of decide further how to analyze these attentions and clusters. And to summarize, I described a predictive model for using the chromatin to predict gene expression using transformers, and then using the attentions of those transformers to kind of identify features and positions in the input chromatin to potentially find interesting or predictive features in the chromatin that drive the prediction. So I described some future directions of analyzing these attentions and incorporating, there's other ways that we can probably decide on how to incorporate other genomic features into the model, such as nucleotide sequence or annotations in the genome, and maybe think about how we can generalize these models to other training and test schemes, other experiments, and maybe think about how higher order organisms can be handled with this model. And that would require longer range ideas like enhancers. But I think this will be an important model to potentially find some insights into the regulatory function of the chromatin. So I'd like to thank the Harding Lab and McAlpine Lab and ISMB for hosting and allowing me to share this work with the community.
Thank you very much for that. We do have an online question that came in. I think it's something you started to hint at in the future work, but we'll let Sean ask it anyway and see if you have any further thoughts.
Okay. Yeah, this is from Myra Fernand-Megaris. So she says, beautiful work, Tron. Do you think this will work as well in mammalian genomes? Also, have you tried to integrate these data with 3D chromatin contacts from high-seed or micro-seed?
I think for higher order organisms, it'll be interesting challenge because of the long range interactions. So right now in yeast, there's no idea. I mean, there's less. You can kind of use a smaller window for as our input for chromatin because there aren't long range enhancer type regulatory mechanisms. So the problem becomes more difficult when we have longer range analysis. And then for I think the second part was like using high C or chromatin context. Yeah, we've thought about different ideas of incorporating other data sets. I'm not too familiar yet with how to incorporate that, but it's something that might be interesting.
Very nice. Thank you. So very nice work. So I was curious. So transformers are supposed to be good for like long range interactions and also attention. I was curious, like if you've actually, I mean, it doesn't look like very long range, like.
Right. Yeah.
Do you need a transformer, and could you have just used a combination of your attention?
Yeah, I think that the idea of using the transformer, I guess, was to eventually be able to extend it to longer range. I've seen like convolutional networks would also probably be useful here, but the idea of using transformers for longer range, and I guess the previous question talked about mammalian or more complex genomes, I think would still be useful.
Can I just ask one other? I mean, in terms of benchmarking, how well does it work in terms of predicting expression?
It works in this testing scheme. It worked pretty well, but we still have work to do on trying to generalize it to other types of experiments and maybe try to hold out a chromosome or hold out other genes. So it's not unclear yet whether it's how the performance is for more general cases.
Thank you. It was a lovely talk. The graphics are beautiful. To your point about the two categories of genes that are highly expressed in different structure, did you find a difference between essential versus non-essential genes when you looked at the function of those?
That's a good idea, but we haven't looked too much into that. We probably should do some gene ontologies on those clusters, and maybe because we do know that there's specific genes that are most highly expressed in this perturbation assay. So it'd be useful to see what clusters have those genes and where they fit. So, thank you.